There we go. That looks good. So thank you all for joining us today. My name's Liam Briley. I'm the chair of the RSS Merseyside Local Group. In this room today, we're not expecting a fire drill, I should say, but there we go. That looks good. So thank you all for joining us today. My name's Liam Briley. I'm the chair of the RSS Merseyside Local Group. In this room today, sorry about that. Right. So if you experienced a horrible chain of echo there, I do apologize. I was saying, if you're attending in person and the fire alarm goes off, then we'll need to just vacate out those doors and out to the fire exit to the left. We're also broadcasting this live from the University of Liverpool via YouTube. So welcome to colleagues and friends who could not be here in person. So today we're going to be talking about modelling approaches for neglected tropical diseases. And some time ago, as a local group, we did wonder about doing an event based around COVID-19 models, and there's some really fantastic research on that. But much of it has already had a big spotlight. So today, I'm very glad to be hosting this event on neglected tropical diseases. So these are infections that are under-recognized, but hugely important, particularly for communities in poverty. And this afternoon, we're going to welcome 4 excellent speakers. So in the first half, Dr. Jennifer Lord and Dr. Jack Pilgrim, they're going to present research on vector-borne viruses. And then in the second half, Dr. Olatunji Johnson and Dr. Claudio Fontere will discuss methods for modeling parasitic worms. So each of our talks is going to be about 20 minutes, and we can take some time for questions following. Before we get started, I just wanted to highlight we do run events on all kinds of statistical hot topics, and our next event is going to be in April. And will very excitingly coincide with Liverpool hosting the Eurovision Song Contest. So we've got 2 academic speakers and a third speaker to come. Later this year, we'll also have an event exploring how AI is used in computer games. And we also hope that these are going to be hybrid events, but do keep an eye out for more details later on. And you can find out more about us on our website, where you can sign up to our regular newsletter to find out more about local maths and stats events, not just ours. We also do have Twitter at RSS Merseyside, and we have our YouTube channel where this is going out live, and it will be able to be watched back on that channel afterwards, along with all of our other previous meetings. We do also recommend that you subscribe to this YouTube channel if you'd like to know when our events are going live. Should also briefly mention registration, abstract submission for the RSS Annual Conference in Harrogate this year is now open. So I think you've got until something like 5th of April to submit talks. And if this is your first time attending or watching one of our events, I should explain that we locally represent the Royal Statistical Society, who are for absolutely anyone interested in data and its uses. So no matter what your career stage, no matter what your field. So we've got specialist sections for medical statistics, environmental statistics, and also data science. So there's more about the benefits of membership, including free e-membership for students on our website. But I think that's quite enough from me. So I'd now like to introduce our first speaker, Dr. Jennifer Lord. Jennifer Lord is a lecturer in vector-borne disease ecology at the Liverpool School of Tropical Medicine. And she also holds an MRC Career Development Award and a Janet Hemingway Fellowship. Her lab investigates the impact of ecology and environment on vector-borne diseases. So I'm going to pass to you, Jen.
Thank you.
And I'll just find your slides for you.
Let me just get this on. Okay. Sorry, one minute. Okay, thank you very much for inviting me. It's nice to be here. So I was going to introduce myself, but Liam's done that very nicely for me. Yeah, I'm based over in the Liverpool School of TropMed. I'm a biologist by training who came to modelling during my postdoc research. So as mentioned, yeah, I'm going to talk a little bit about sources of mosquito-borne viruses that have hosts primarily in animals other than humans, and for which humans are sort of dead ends, but which where disease occurs. Using 2 case studies, Japanese encephalitis virus and Rift Valley fever, with the main focus on Japanese encephalitis virus. So just to give a quick overview, there's quite a bit to cover in 20 minutes, but I've put myself a timer on to make sure I stay to time. So we'll see how we go. I'm just going to give a quick background to me and what my research focus is. And then in the talk, I want to kind of focus on 3 key themes that have been important to me. I think they're probably important to a greater or lesser degree in a lot of disease systems. Those are heterogeneity, the scale of things, scale at which things occur, and at which we want to control diseases, and also noise in the system. And as I said, just want to try and then sort of think about those things conceptually, a bit abstract, and then apply those to the diseases we're currently working on with some examples. So our group, I did a postdoc. I've been at LSTM since 2015. I did my postdocs there, and then started my own group in 2020. So we've got a bit of momentum now in the group, focusing on these two main projects on zoonotic disease. And as Liam mentioned, so we're focused primarily on disease ecology, and also with a focus on how anthropogenic change, land use change, livestock systems change might be influencing these pathogens and the resulting diseases. And just as highlighted on this slide, I'm an ecologist and biologist, and I'm really interested in how things interact. And so generally speaking, as you add things into a transmission cycle for a pathogen, things become obviously more complicated, and also more dependent on context. So we know if you have a mosquito, a vector, they are dependent on environmental temperature, on hydrology, and other aspects of a local environment, thus making differences across time and space. So you often will get focal outbreaks and quite a lot of variation in transmission across the landscape. So it's the cycle towards the end of this schematic that we're particularly interested in, where we have these multiple hosts and often multiple vector species. And so why do we care about ecological context? So our group's really interested in how ecological and environmental context influences both the vector population dynamics and those of the pathogen. And if we can actually leverage the knowledge we generate about the influence of context to help target control. So often resources are limited, and if we can target control in a way that's more efficient, then that should be beneficial and more effective. So as I mentioned, I came to modeling slightly later, and my postdocs have been focused on real sort of field work, thinking about some of these big questions, how local environment influences transmission. Mostly previous research has been on this, the tsetse fly on the right, which is a vector of the parasite that causes sleeping sickness. But then our group now is predominantly focused on mosquito-borne viruses, and this is Culex tritinea, which is the primary vector across Asia of Japanese encephalitis virus. So that was a quick flavor of our group and what we're doing. And I want to just talk about these 3 key themes in the disease systems that we study in our group. And so here's a schematic with lots of things going on. So it's been quite widely acknowledged for a while now in vector-borne disease modeling that there's lots of complexities that should be better accounted for. And this has been done quite well for some disease systems. Everything from variation in larval sources for mosquito vectors, through to what the adults are doing, their behavior, the hosts they're feeding on, and then host responses and how these things interact to drive transmission. And in particular, there's been some nice work for, particularly for pathogens with humans as the only host, primarily, so dengue, malaria, in most circumstances where vectors will only feed on humans, that have looked at how spatial variation and think how populations are structured in space influences transmission, and essentially sort of demonstrating how human movement really drives transmission and can explain variation in risk in urban areas. But as soon as you add in other hosts, things haven't been as well studied. So here, the schematic here just outlines the sorts of parameters, so the aspects of biology that we often will add in models of pathogen transmission, the zoonotic mosquito-borne pathogens for viruses. So on the left is what happens inside in the mosquito population. So some important aspects of mosquito density, how long they live. And on the right is what's happening in the hosts, how much virus they produce, host density, their turnover. But the bit in the middle is what's particularly interesting is these interactions between hosts and vectors. And so when you add in different host types, you have some hosts that can transmit pathogens and some hosts that are a dead end. And also you have vectors that will often, by necessity, if it's a zoonotic disease, they must be feeding on wildlife or livestock and humans, which adds in an extra level of complexity in the disease system that hasn't yet been fully accounted for in modelling efforts. So the way that we've been thinking about this is we know that zoonotic arboviruses are particularly heterogeneous. Their outbreaks are quite unpredictable. Where is riskiest is difficult to determine, and we're thinking about how can we best get a better understanding of that to target control. So we're starting to think about things in terms of source sink dynamics. So this is a well-studied area in ecology generally. So imagining patches in a landscape that may be a source of transmission, and areas which may be a sink and can't actually by themselves sustain transmission, and how easy it is to identify these sources and how important and how will this influence how we target a control program. So we can also think about sources across different scales. So at the individual level, one of the important things to do is identify what host types and what vector species are important for transmission. So you can imagine a competent host, maybe a potential source at the individual level, and also a vector that's able to become infectious with a virus is also a source at that level. And then you can think about at the next scale, the level at which host vector contacts occur. And in some scenarios, this might be, you can think of this as a household level. So if there's a household that have some livestock and some humans, and the mosquitoes come to individual households, they can vary in their livestock community. You could also think about it in terms of patches in a landscape that maybe support birds and different animal communities. So we add this, in this layer, we can think about how host community composition causes heterogeneity in the system and influences the host within which mosquitoes take a blood meal. So these things combined together will determine which species contribute most to transmissions. That's one of the things we're interested in to answer for Japanese encephalitis virus. And then also at the scale above households, what influence does this sort of host vector contact heterogeneity have at population level within villages or between villages in terms of transmission dynamics and sources and sinks of transmission? And what we're interested in at the moment particularly is, as I mentioned, how does this heterogeneity influence the proportion of blood meals on competent and dead-end hosts? And how does this vary in between households and between contexts that have different host communities? And so this is an extreme sort of example of this. So you could model a vector-borne disease in a system at the top where you have host cattle here as a dead end and birds as an amplifying host of the virus. And if they're equally distributed and a vector prefers cattle, you can imagine it being difficult to sustain transmission. But actually, if you ask the question, well, if we separated that population out into discrete patches, some of those patches may have many competent hosts which are fed on frequently, and actually can those patches sustain transmission? And if you start to then have in between, because obviously in reality it'll be an in between of those two scenarios. How does that play out for transmission? And then the question, why do we care about that? I mean, it's biologically interesting, but is it useful? And one of the things we want to know is, can we leverage knowledge at that level to help target control, but where data available may be only present at a higher scale? So at what level would additional data about livestock community composition or host community composition influence our ability to think about where is riskiest for transmission? And is host and vector community composition predictive of risk at these larger scales, or is actually just too much noise in the system? So we know that there's lots of things that affect arbovirus transmission, and maybe actually even if we get a really good understanding of what's happening at household and village level, we still might find it difficult to predict where is riskiest and be able to target control. So the projects we have at the moment are addressing these questions at different scales. So again, I've sort of gone quite quickly in 10 minutes through these three themes about heterogeneity and scale and noise. I'm going to try in the next 10 minutes to just show how this is being applied for Japanese encephalitis virus. To hopefully just give it a bit more context and how this actually might look in reality. So we have two main grants in the group at the moment. So as I mentioned, one is on what are the drivers of Japanese encephalitis virus specifically in Bangladesh. And this is the project I'm going to focus on in this talk. I don't think I'll have time to mention about RIF, but I wanted to put it up there. We have got another project it's really similar in concept sort of theme, and that's on intraepizootic transmission of Rift Valley fever virus in Tanzania. So if anyone has any questions on that project, I'm happy to chat about that. So Japanese encephalitis virus, so as hopefully you'll have assumed, it's a mosquito-borne virus. Its main hosts aren't humans. It's present across Asia, and it's a neglected tropical disease, so burden is underestimated. The last approximate estimates, I think, are from 2015, where there was an estimated 25,000 cases, despite the fact that there's a human vaccine and there's national vaccination programs in many countries. There was a really nice study a couple of years ago by Kwan et al. That showed the impact of vaccination. And the important thing from this figure here is you can see in some countries there's no change in colour, so Bangladesh is the same colour. So there's no vaccination program in Bangladesh yet. So our project is hopefully helping to will help to build on the informing how that vaccination program gets deployed. It's going to be in the next couple of years. And then the other important aspect of this figure is even in some countries where there have been national vaccination programs for a while, in particularly India, the anticipated effect of vaccine hasn't been achieved due to a combination of coverage and efficacy. So again, seeing that even when we do have a control method, improving ways of improving coverage in riskiest areas would be beneficial. And then we could, so yeah, we've sort of hopefully get a feel for, we want to be able to know where is riskiest for these sorts of pathogens. And you may think, well, we should just go where there's human cases, but these pathogens are often rural diseases. And there was a study by Nicolai et al. a few years ago that showed the probability that you actually detect a case of Japanese encephalitis virus and some other pathogens, it drops off substantially as you move away from hospitals. So you might have human surveillance, but those human cases might not reflect where is actually at highest risk. So what other methods can we leverage, and can we use data on host vector interactions to help sort of pinpoint these riskiest areas? And then the question also is, if we want to know where is riskiest, we also want to know what's driving transmission. So as I mentioned, this is what we think the life cycle is of Japanese encephalitis virus. It was first studied in the 1950s in Japan, where pig production was the main livestock system. And so pigs were implicated in transmission, but there was also a role for ardeiid birds, which often frequented rice fields in Japan where the mosquitoes breed. But that system is really, everyone really kind of assumes that this is the main transmission system. And this is what an area of Japan looked like in the 1950s in terms of its livestock. So each square is 10,000 animals. So you can see there was very few ardeids. Poultry, they would have had some, but I think it was kind of minimal. Cattle, which are dead end hosts with Japanese encephalitis virus, were very few, and pigs were quite numerous. If we take this over to Bangladesh, so if we think about an equivalent area in Bangladesh, this is what its livestock community looks like. So it's a little bit different. The first thing you'll notice is there's very few pigs. So when I went there, which is more than 10 years ago now, my immediate question is, well, how on earth is this virus even persisting? The other important thing that we know is the mosquitoes that transmit JEV really like to feed on cattle. And there's lots of cattle in Bangladesh, so you would think that there's just a massive sink for virus and it's not going to go anywhere. But then obviously in contrast, we have lots of domestic birds, ducks, pigeons and chickens, but we actually don't know what role they have in transmission across Asia. So part of the project is geared to address that. So yeah, this is just as I mentioned. So I spent 6 months in Bangladesh a decade ago now. But when I was there for 6 months, this is the only pig that I saw while I was there. So they were really rare, and I just couldn't get my head around how this guy was maintaining this landscape. So I've made these points. So we know that the vectors prefer cattle, and cattle are numerous in Bangladesh. But we know that birds can produce sufficient viremia to infect mosquitoes, but we have no idea what contribution these are making to the transmission cycle, and we have no idea, importantly, how often vectors are feeding on birds. So we have some field work to start to look at this. So the main questions for this project, for both the field and the modelling, is how do epidemics occur with a high density of dead-end hosts? We also want to know which species contribute most to transmission. Can we leverage that knowledge to target vaccination efforts? And so back to this schematic, and one of the things I wanted to, 5 minutes, thanks. One thing I wanted to make, one thing that I've learned to do while sort of coming from a biology background, but thinking about things mechanistically, is bringing modeling in at the start of a program. So we have this conceptual model here. So most mathematical models can be fleshed out from something like this. You start with a figure of the system, which encapsulates the basic parameters, or the most important things you want to model. And so our field work and our hypotheses were sort of driven by this, with a focus on the things that we thought would be most important and would vary most. So this host vector interaction and also host densities. So this is a mess. And I don't want to go into this, but it's just to highlight our approach in the group is multiple hypothesis testing. We don't have a single hypothesis. We accept things are complicated in the real world. So this messy system is what we're focusing on and trying to get a handle on. But we want to focus in on what's happening here. So which pathway? So you can see these as pathways to the bottom of a human being exposed. We're focusing on this pathway. Which one is most important? So all of our modeling efforts are geared towards designing field experiments that can test these multiple hypotheses, which will be represented in a set of sort of different models that vary in how they represent this system. So using causal inference. And then this is the planned set of studies that we designed based on sort of a conceptual model and thinking about things mechanistically. And again, I haven't got much time to go over this, but what I wanted to just focus on is we've got these village level studies to think about which birds are fed on most, and how does mosquito preference and host choice vary with host variation within household levels. We're starting that this year. To answer this idea of which hosts contribute most, you might have some circumstances where you have lots of chickens. We know we've got ducks in rice fields, then we've also got lots of cattle in between, and how is that influencing transmission? And at the same time, we're building these models to help explore theoretically. So again, there's not much time to go into it. So just to give you a quick flavor, we're thinking about things separated out into patches, metapopulation models, where each patch has a different host community composition. And then we can take host density in each patch and decide, okay, what proportion of hosts are likely to be fed on in each patch? And then how does that then play out for transmission dynamics? And just to show you, again, it's sort of giving a flavor of the sorts of results we can see is the plot on the far left is if we imagine a homogeneous, all the hosts and vectors are mixed up equally, and there's equal numbers of dead end hosts and competent hosts. And then we've just run a simulation model out over a year, and the y-axis is the prevalence of virus in mosquitoes, and you can see in the dark blue, where fewer mosquitoes have a lower preference for birds, you actually don't get any outbreak, or there's very little transmission. But then if you start to divvy out the hosts into two patches, where you have more dead-end hosts distributed, so this is now in the far right, more dead-end hosts in one patch, so they're sort of aggregated, you can actually get an outbreak in these circumstances, even when you have relatively low low preference of mosquitoes on the competent hosts. So I apologize that I've rushed through that a little bit, but happy to kind of answer questions. So essentially thinking about things in metapopulation level. And so what we want to be able to do eventually is think about things at a larger scale. So how does community composition vary at a scale at which we have livestock census data? Is it predictive of JE risk? Can we, if ducks are important for transmission, for example, can we target areas where ducks are relatively common? How much variation is there at a union level? So that's like a cluster of villages in livestock keeping. And what we're actually going to do in the project, importantly, is test our models. So we have a large livestock cross-sectional study in the 3rd year to test whether our models can predict the areas that are riskiest, and essentially using cattle exposure to virus at 0 prevalence in different villages. So quickly to summarize, I've given hopefully a little flavour of the heterogeneity we're starting to think about, and how can we identify hotspots of transmission? Can they be targeted for control? And for that, we need a better understanding of local host vector interactions. Scale comes up all the time, and particularly also in designing our field studies. So how are we going to sample mosquitoes? How are we going to think about data from the hosts, and at what scale? And in terms of control, that always happens at quite a local scale, but the data to inform that is normally an aggregated, a higher scale. And then the other thing we're dealing with is we're focusing on only a couple of aspects of the system, and there's always going to be things that we can't account for, and how much is that going to cause a problem? Is there going to be any signal in the sorts of things we're looking at? Or is it just too messy that we can't actually predict where and when these pathogens occur? Thank you.
Thanks so much for that, Jen. Do we have any questions? And I should say as well, if you're watching on YouTube, we can absolutely take questions through the chat. Could I pinch the microphone?
Yes.
Are there any questions in the room to start off with? Yes. Sorry, I'll just pass you the microphone.
To what extent is the proximity of the host, the animals, to the human populations important? Because from my memory, mosquitoes pretty lazy, don't fly very far. So to what extent is that a factor, a compounding factor in that? If you've got houses where the hens and the cattle walk freely among and even come into the houses, that's going to be a different dynamic to if the houses are kept separate from the host.
Yeah. So it's important, and so one of the things to point out is that we've ignored the actual step between mosquitoes and humans in the project, because it's a whole different, it would be a whole new project in thinking about human risk. So at the moment we're thinking about drivers of transmission and where's riskiest in terms of infectious mosquitoes. But there's definitely data to show, particularly for malaria, influences of having cattle in a household versus cattle away. And I know there's studies to show if actually you put livestock between larval habitats and households, you can sometimes reduce transmission because you're sort of intercepting them. Whereas if you bring livestock into the household, there's a push-pull because you're actually attracting more mosquitoes to the household. You might have more feeds on cattle, but and again, I could talk about for half an hour on this, because when you start thinking about host vector contact, it gets really complicated. And there's some interesting interactions between mosquito density at that scale, host preference, and the host they finally choose. So the answer is it's complicated.
Thank you. Do we have any more questions? Okay, maybe I'll just ask a very, very quick one myself. So, I mean, you mentioned like the sheer complexity of these systems. That makes me think of things like machine learning as a possible model method. Like, do you have to be really discerning about the methods that you choose because there is just so many sources of variation?
So our methods, my approach has always been to model the system using a mechanistic approach. Just because I like, so you're sort of representing a series of hypotheses in a mechanistic model. I think you could probably, if somebody was an expert in machine learning, they could address the question in probably a very different way. Yeah. So
Sorry, thank you. Okay, let's thank Jen once more. Now I'd like to introduce our second speaker. So our second speaker is Dr. Jack Pilgrim. He is a postdoctoral research associate at the University of Liverpool, based in infection, veterinary and environmental sciences, where he was awarded his PhD in 2020, near enough 2020. He's a qualified vet and his research interests cover veterinary entomology and vector biology. So I'm going to pass the mic to you, Jack, and I'll find your slides.
Great. Oops. Is it possible to get a pointer on? Yes.
Yes.
You see that? Okay. Well, thanks for that introduction, Liam. It's great to be here. So my presentation actually follows on quite nicely from Jenny's, as far as it involves mosquito-borne diseases. As Liam's alluded to, I'm a veterinarian by trade, but actually some of my research over the past few years is involved in insect vector biology research. And this is actually the first project that I've been involved with that has a modeling component. So if I don't explain certain concepts perfectly, I do apologize, but either way, I hope that you can gain something from this talk. The title of the talk is Usutu virus, a new vector-borne threat in the UK. So Usutu virus is a lesser known virus spread by mosquitoes. And the reason we're interested in it, about it in the UK, is because in 2020, it emerged here. But because it's a new entity, we're not actually sure about the risk it poses to animal and human health. I think I'll just hold it. So Usutu virus is an emerging vector-borne disease, and there are many drivers for these kinds of diseases. The first one is globalization. So this has allowed for the ease of transport of animals, humans, and insects across the world through things like trade and tourism. Urbanization, which has allowed for new habitats to form for mosquitoes and a large wealth of hosts to feed on. And also climate change. So we're now seeing insect vectors in parts of the world where we previously haven't seen them as a result of changes in temperatures and hydrology. In terms of notable historic outbreaks, one of the first on record was in the mid-18th century. So dengue virus spread through North America and Southeast Asia as a result of the slave trade. But at the turn of the 21st century, these vector-borne disease outbreaks seem to be becoming more and more common. So West Nile virus spread across North America and caused neurological damage in both humans and horses. Closer to home, blue tongue virus was spread by biting midges, and this caused great economic damage to the livestock industry. In 2015, Zika virus, which was previously seen as quite a benign virus, isolated in Africa, made its way to the Americas and caused a great deal of damage, especially to newborn infants. And in 2020, we found Usutu virus in the UK. I'll go on to talk about this particular virus a bit more now.
Close that preview. I think I'll sabotage you a little bit there by opening that at the top, just so that people will be able to see.
That's okay. So Usutu virus is an RNA virus spread by Culex mosquitoes. They predominantly transmit the virus to birds, specifically passerines. So these are things like blackbirds and sparrows, and they can cause quite severe symptoms, so paralysis, multiple organ failure, and ultimately often death. So humans are dead-end hosts. They're not very often infected, but they can cause symptoms such as fever and rash, and in immunocompromised people, they can also cause more severe symptoms like swelling of the brain. In terms of epidemiology, Usutu virus was first identified in Mozambique in the mid-20th century, and since then there have been regular incursions of Usutu from Africa into Europe through migratory routes of birds. Through genetic data, we know that there have been at least 3 independent introductions of the virus into Europe. And from around 2000, the establishment of Usutu virus spread from the Mediterranean basin all the way through continental Europe. And in 2020, dead birds were discovered in Hyde Park, London, which tested positive for Usutu. So what do we know about Usutu virus and the UK? Well, it's been detected for 3 years in a row now, so it suggests that the virus is here to stay. local transmission is likely. So from the dead birds which were discovered that tested positive, this was over several weeks. So far, it's only been detected in the southeast, but that's probably as a result of surveillance bias. Aside from birds being infected, we've also found that Culex pipiens, the main vector in Europe, has also tested positive for Usutu around London. So what sort of questions does my project aim to answer? Well, the first is, can Culex pipiens spread Usutu in other parts of the UK, apart from London? Which mosquitoes can act as bridge vectors? So these are vectors that can feed both on people and birds, and so these could potentially be of public health significance. And finally, what is the risk of Usutu spread for current and future climatic scenarios? So you can imagine if Usutu can spread readily in London, but it's not possible in Scotland currently, in 60 years' time, will that scenario change? So when we're considering modeling the transmission risk of vector-borne diseases, there are a few variables that we need to look at, specifically related to vectorial capacity, which is the general ability of the vector to transmit the pathogen. So the first one of these is biting success. So just to note here that a mosquito needs to bite twice in order to perpetuate infection. The first bite obviously gains the viremic blood meal, and the second transmits it to another vertebrate host. And this is dependent on the number of available hosts, which you can get this information from field data. In terms of transmission competency, mosquito and host immunity, how well can the insect and the vertebrate host clear the infection? This is sometimes dependent on prior exposure to the virus or vaccination. And intriguingly, the microbes harbored within the insect itself can also affect their ability to transmit viruses. So the most well-known example of this is Wolbachia, which is now being used to reduce the burden of dengue fever in the tropics. Aside from this, we have daily survival of the mosquito. So obviously this is important. The mosquito needs to survive long enough to take those two important blood meals. And extrinsic incubation period, which is effectively the time it takes from a mosquito taking an infectious blood meal to becoming, a blood meal with virus in to becoming infectious itself. And these two variables are importantly affected by environmental temperature. Generally, the higher the temperature, the shorter the lifespan of a mosquito and the shorter the extrinsic incubation period. And if you flip that on its head, the colder the temperature, the longer it will take to overcome the extrinsic incubation period, which means that there will be a critical lower threshold temperature for virus transmission. And this is a particularly important concept when we're thinking about transmission risks to temperate regions, like in this study. So how do we understand more about these parameters? Well, we can use two different systems. We can use field caught mosquitoes, or we can use colonized mosquitoes. So these colonies have usually been in culture for several years or even decades in the lab, and there are pros and cons to each system. So for field caught, these are actually representative of the population of interest. So these mosquitoes we're bringing in and experimenting on, these are the ones that are putatively able to transmit whatever virus we're looking at in the real world. However, there are drawbacks. So these field mosquitoes are only available in certain months, mainly in the summer. And there are other logistical challenges like finding particular breeding habitats and also finding rare species to work on. In comparison to a colony where numbers are available all year round, it's good for replicates. So if you imagine I go to a field site here in Liverpool, bring them back to the lab and do an experiment on them, I want to replicate that data. I go back to the same site a year later and the population will be different. Whereas with the colony, that's not the case. They're also lab adapted, which means that they can reproduce and survive quite more easily in a lab compared to field caught mosquitoes. But importantly, they are often genetically and behaviorally different to field mosquitoes, and so they're not quite representative of our populations of interest. So I'm going to use field caught mosquitoes for this particular project to understand the ability of Culex pipiens to transmit Usutu alongside this list of bridge vectors of interest. And I'll briefly go over the workflow here. So I'll go to various urban and rural sites, find habitats, mainly for Culex, bring back image for stages to the lab, rear them at ambient UK temperatures. I'll then expose them to Usutu and incubate them at various temperatures. And then at different time points, I'll then sacrifice them, but before doing this, I will knock them out with the chemical and place their proboscis in a capillary tube with mineral oil, and this will make them salivate. And I'll then take the saliva, do an RNA extraction on them, and use a technique called QRT-PCR, which is effectively a molecular method to detect small amounts of genetic information, so understand whether or not the virus is present in the saliva or not. And hopefully I should get data that looks like this. Currently, I'm only a few months into this Usutu project, so I don't actually have any data to show you per se, but this is data from my current group, which I'll elaborate on a bit later. So on the y-axis, we have proportion of saliva positives, and on the x-axis, we have days post-infection. So how do we get from this empirical data to risk maps? So if you look at the graph on the left, we can glean two bits of information. The first is minimum temperature necessary for infection. So if you look at the two lines at the bottom here, 17 and 19 degrees, we don't see any mosquitoes becoming infectious during the time course for 17 degrees, but we do see some at 19 degrees, which suggests that this minimum temperature necessary for infection lies between these two temperatures. And the other is extrinsic incubation period, and I'll elaborate on that in a second. But hopefully, adding this into a model will create the risk map that you see here. So as I don't have my own data to present, my colleagues have kind of allowed me to present their data, which involves actually Zika virus. But I intend to use very similar methods, so it's quite a good case study. So the title of this study is the potential for Zika virus transmission by mosquitoes in temperate climates. So as I mentioned at the beginning, Zika virus became a problem in 2015 in the Americas. It caused the birth of infants with very small heads, otherwise known as microcephaly. And this led to lifelong disability for these children. So in terms of differences between Usutu and Zika, Zika is a different vector, the Aedes mosquito. In this particular study, they used Aedes albopictus, which is present in temperate climates, but actually the primary vector is Aedes aegypti, which is usually found in the tropics. The host is humans, and the distribution varies. Now, the reason it's important to note that there were such severe outcomes of Zika infection is that previously Zika wasn't seen as that important in terms of mortality and morbidity until 2015. And so a similar thing could happen with Usutu. At the moment, it's not seen as being a particular public health interest, but who's to say in a few years that it might become just that. So these graphs on the left here are similar to the graph I showed you a couple of slides ago. The only difference is a logistic regression model has been fitted to the data. And this allows us to calculate the IP10, which is the time period it takes for 10% of the population to become infected at a given temperature. And this can be plugged into this model on the right here. So some of you may be familiar with the concept of R0, which is the basic reproductive number. It's an epidemiological term used to describe the number of new infections in a completely susceptible group once the disease is introduced to that population. So in this context, it was actually modified to be a standardized R0. So the value was between 0 and 1, and that was to fall in line with previous methods used in similar work. So in terms of the other parameters, a lot of these are temperature dependent, so biting rate, mortality, and extrinsic incubation period. And some are non-dependent on temperature, so host recovery rate and vector-host ratio. So a lot of the values, these parameters were gained from other studies where they've looked a bit more closely at things like biting rate and mortality at different temperatures. So the importance of using standardized R0 is that it can be used as a proxy for transmission risk. So using the example here, Aedes albopictus, infected with Zika virus, the minimum temperature threshold lies between 17 and 19 degrees, which corresponds to a standardized R0 of between 0.2 and 0.3. And if we plot standardized R0 here, we get this curve. And the beige area in the curve represents this range, which also represents possible transmission of Zika, but unlikely. Anything above 0.3 is possible transmission of Zika from Aedes albopictus and likely. So if we use this color code to look at current global risk based on recent climatic data, we can see that a lot of risk for Aedes albopictus and Zika virus is in the tropics and Africa, as would be expected. One thing to note is that a rainfall criterion has also been added to this model. So places like the Sahara Desert and Central Australia are masked because these areas can't allow mosquitoes to proliferate. So I just want to draw your attention to two things on this plot. So the first is in North America, where we see moderate risk for transmission. And what's interesting is that we know the Zika virus has been locally transmitted in places like Florida and Texas. And we also know that Aedes albopictus exists there. So this mirrors what the model predicts, which is good. If we look at Australia, there's moderate to high transmission in northern Australia and Queensland. Now, mainland Australia doesn't have Aedes albopictus, and we've not seen Zika transmission there, but it does harbour Aedes aegypti, which is a different vector. So it could be the case that this information could be used for targeted surveillance of Aedes albopictus and Zika still. So moving on closer to home, this is a map of Europe with the dots representing Aedes albopictus presence. Most of them are in Italy, South France, Sardinia. And alongside this, we get stippling of beige, which suggests low transmission. But again, around the time that this project was published, there was local transmission observed near Marseille, and again, showing that this could actually be quite a sensitive model. In the north of France, we also have Aedes albopictus, but this shows no risk of transmission, which also suggests that the presence of Aedes albopictus alone isn't sufficient to say that there will be transmission. So looking at different climatic scenarios, we have the largest increase in R0 in temperate regions in the south of France, eastern Spain and Italy, but not the UK, which basically suggests that the UK currently and in the future won't be at risk from Aedes albopictus transmission of Zika. So what lessons can we learn from this Zika example for my project? Well, they used a Aedes albopictus colony, which is understandable considering that albopictus doesn't exist in the UK. I of course will use field collecting mosquitoes, but as I'm investigating the smaller biomass, i.e. the UK, I'll be able to take transects of the UK and go to different sites to collect different populations, which might vary as a result of temperature or unknown factors. Second thing I can do is narrow the range of incubation temperatures. So obviously, globally, temperatures are quite wide, but that's not the case in the UK, and also means that I'll be able to have higher resolution between these temperatures. And the Zika virus project didn't allude too much to presence or absence of humans, which is fair enough considering that we're ubiquitous across the globe. But as we know that we have particularly birds that are likely to be infected by Usutu, I want to include a parameter involving presence and absence of birds using information gained from the British Trust for Ornithology and the Royal Society for the Protection of Birds. And finally, in terms of specific challenges for this project, it's easy enough to get Culex pipiens from the lab, from the field into the lab, but it's really difficult to get them to feed for various reasons that I won't go into. But I'm hoping to use certain phagostimulants, which are basically chemicals that will encourage feeding to try and bump up numbers so I have sufficient sample size for these infection experiments. So cryptic species identification. Cryptic species are basically different species that can't be identified morphologically or by eye. You have to use molecular methods. So that's another thing that will take time in order to differentiate which species are which. In terms of acquiring accurate parameter information from the model itself, For Aedes albopictus, this is a well-studied organism, which means that we have a lot of information in terms of its biting rate, mortality, etc. This is not the case for some of the organisms that I want to use in the current project, and I may even have to do my own empirical studies to get some of these parameter values. And finally, sample sizes for field cover mosquitoes are difficult. So if you imagine hundreds to thousands of adult females are needed to create sufficient numbers to undergo experiments for various temperatures and various time points, and that actually the mosquitoes you bring into the lab often will die, or will be males, or will be at different ages. It's quite difficult to ensure that you'll get sample sizes that you want. So for this reason, as I'm going through the project, I may need to prioritize particular temperatures or particular time points. And so to conclude, temperature has a big impact on transmission and can be manipulated in the lab to model transmission risk, as we've seen with Zika virus. Empirical studies can be improved to give better validity to models, especially bringing in mosquitoes from the field. However, challenges include logistical issues of getting them to feed and finding them in the first place, as well as limited ecological information for understudied putative vectors. So finally, I'd like to acknowledge the people that actually did the work that I presented today. So that's Soren, who did the modeling, and Marcus, who looks like the incredible hawk on a bike there, who did the lab work. And I'd also like to thank my funders, University of Liverpool for hosting, and yourselves for listening. Be happy to take any questions.
Fantastic. Okay. Do we have any questions for Jack in the room to start with? Oh, yep.
Do you know if is there any, so as these viruses have spread, so obviously climate change can influence spread, but is there any data similar to like West Nile that like Usutu virus has changed and adapted to temperate? Do you know if is there anyone who's looked into that, like evolutionary dynamics of
Not that I'm
Usutu?
aware, there's not.
Sorry.
So the literature on Usutu is still quite limited. As Usutu's gone through continental Europe, you'll see the year of publication of when it was found in that particular region. But they're mainly just trying to figure out prevalence and very basic epidemiological information. So no, we're not aware at all about that kind of stuff, unfortunately.
Have we got more questions? Yeah, let me just bring the microphone over.
Thank you. Yeah, I was just wondering how you've selected your temperature range of 17 to 23. It seems like quite a small range, and it could be warmer than that in London, I was thinking. So just interested in that.
Yeah, thanks for the question. So yeah, you're right. Of course, it can get hotter than 23 degrees. So at the moment, these models are using average daily temperature. So it's unlikely that even in sort of the worst case scenarios for emission projections that will have average temperatures over 23 in London, perhaps. But we certainly know that the lower critical threshold could be lower than 17. So I think it's West Nile virus that can still be transmitted at 14 degrees. So if anything, I have to probably go lower, I think.
We've got time for another question if there is any. If not, then let's thank Jack once again. So what we'll do now is we'll have a 10-minute break and we'll resume with our second lot of speakers. After that, I make it just about 3 o'clock now, so we will stop for 10 minutes. I believe there should be some tea and coffee in the room, and if you are watching online, then please do rejoin us at 3:10. Thank you. Okay, so hopefully we should be back online now as well. So now I'd like to introduce our third speaker, Dr. Olatunji Johnson. He is a lecturer in statistics in the Department of Maths at the University of Manchester. He obtained his PhD from Chickas at Lancaster University, working with Peter Diggle, Emmanuel Georgi, and Joe Knight. And his research focuses on developing novel geospatial methodologies for epidemiological statistics. So let me find your slides, Olatunji. Oh, they're on this. There we go. I'll pass the microphone to you. Are you okay with getting it full screen? Fantastic.
All right. Hi, everyone. Good afternoon. My name is Olatunji, and today I'll be talking to us. I'll be talking to us on statistical method for efficient safety assessment of COVID-19 in rural and endemic areas. I'll start by showing us these two maps. The one on the left is the oncocensus map, while the one on the right is the rural map. So basically, these are early maps in the early 2000, I'll say. That's when they created these maps. And what happened was that they wanted to eliminate oncocensus. But in eliminating oncocensus, which is also called river blindness, they found out that in some areas, people are co-infected with lower and oncocensus. And those people, by the time they administer information tablet to them, they suffer serious adverse events. So this was early map. So they found out that in areas where there is high risk of both diseases, people suffer serious adverse events. And that is why it is important that they identify areas. So in order to eliminate oncocensus, they need to identify areas where there is high risk of lower, so that they don't administer mass drug administration in those areas. So that is the essence of my talk. So my talk is basically centered around trying to identify areas of high risk of lower for efficient MDA, mass drug administration. So the map I showed earlier, the way they collected the data, you might be wondering, how did they produce their data? So basically what they do was to walk around villages and ask them questions. That is called RAP-LOA. They are basically a set of questions. Just show you a picture. You have this kind of thing in your eye before, this kind of things. And then they're able to come up with some numbers, and then they use that to produce their map. But that was the early maps. But things have advanced. Now they have, they could develop more sophisticated things to probably come up with data for mapping purposes. So there's a lower scope, which is also developed very recent anyway. So this basically what they do is to go into the village, they collect blood on this smartphone device, and then you'll be able to measure the microfilarial density in the blood. So this one, and then I'll show you the last device as well, which is the antibody test, which is cheap. So there's antibody tests too that they could also conduct, use that just test people. So the problem with lower scope is that there are very few lower scopes. And carrying out, if you want to imagine you want to go to a country to come and carry out MDA, you need at least reasonable number, and there are only about 100 lower scopes available. I don't know if they have produced more now, or at the time I was doing this study, there were just about 100 available. So it's not really enough, and that's why they have to supplement it with an antibody test. And now they're asking us again. So the question they asked us was that we don't have enough lower scope. How can you help us combine the information from the lower scope and the information from the antibody to identify high-risk areas of lower? So this study was conducted in Gabon, and this is the map of the southern part of Gabon. So the shaded regions there, they are the districts, you can call them districts, but they call it departments in Gabon. The dots on the maps, they are the villages that were surveyed. and they went to, and then the red partition, you can see the red partition on the plot. Those are, I call them pseudo enumeration units, pseudo EU. And the reason why I've done that, I'll tell you later during the course of the talk. But what I've done there was just to, you can just think about putting ruler on the map and just drawing lines. Basically, just to make sure that you have a population of about 5,000 to 15,000 in each pseudo EUs. So let's go into the data. So as I've told us, we have the data on antibodies. So we go to the villages or collected data on use antibody test and use lower scope test. And what lower scope test we measure? So antibody test, that's Y1. It's just basically bioreactor. Is it positive or negative? The lower scope we measure the density, microfluidic density in the blood. So those are continuous outcome, really nice. And then what we did was to create Y3 there from Y2. That is, if the microfluidic density, if it's greater than 150, if it's greater than 150, we say that's positive. If it's less, then it's 0. So basically, we created Y3 as a binary outcome. And the choice of 150 is based on some sensitivity analysis that we did on the device. So basically, we want to combine these 3 data sets to identify areas that are safe for mass drug administration. That is our research area. So I'm just going to tell us. So the method, the way we approach this method is we use joint statistical modeling. And why do we choose this method and what are the advantages of this method? So the good thing is that the first thing I will say is that this joint modeling basically allows us to borrow strength from all the outcomes. That we can explore the correlation between this Y1, Y2, Y3 that have showed us, those 3 data sets. And another beautiful thing is that, which is very peculiar to geostatistics, is that we only sampled very few villages, and we want to make inference about the villages that were not sampled. So we could use geostatistical approach to borrow strength from the villages that we've collected data to predict for the villages that we have not collected data on. And that's actually respect the first law of geography. That is, things that are close together, they are more related to things that are far away. So we can borrow the correlation to predict for unobserved villages. And lastly, which is very peculiar to most modeling, most models that we develop, is that you can use small data sets to actually make inference. You don't need to say, okay, we want to carry out sensor, because that is even more cost efficient. So you can use limited data sets to train this model and then make inference. So let's go into the equation, a little bit of the question. So this is our data model. Basically, what we're doing here is just assuming distribution for all our data sets. The Y1, which is the binary outcome, we impose a binary distribution on that, with probability of positive as denoted as rho there. Then the Y2, so basically the Y2 given Y1, if Y1 is positive, we know, if Y3 is positive, that is, if Y2 is greater than 150 microfluidic density, we model that as Weber distribution. Weber distribution is just a continuous distribution. Of course, one could consider any other distribution here. In our choice of Weber distribution, we also consider the gamma distribution, but we found that Weber distribution did better job for this data set. And you can parameterize that distribution with the scale parameter and the shape parameter, lambda and gamma there. And the Y3, we also assume a binary distribution. So this is our model structure here. We can see those 3 models, they're just the 3 set of data that I've showed. Well, will tell us what's on the right-hand side of that model. Basically, you can see the mu1, mu2, mu3 that is common to them. So those are the fixed effects. Basically, they are just the way we've accounted for the things that we know about the duration of these diseases, this outcome that we have. So the things that we know. So we modeled that using EVI, enhanced vegetation index, and then the elevation. So basically, we used elevation and enhanced vegetation index as covariate in our model. So I'm going to talk about the other term. Let me talk about X naught. So we have X naught, X1, X2 there. So X naught there, basically, you can think about that's the random effect, and these are the things that we don't know or that is unobserved that I want to account for in our data set. So what we did was to model that using Gaussian process with mean zero and some parametric covariant function. So basically, I don't want to go deep into what we did in modeling that, but essentially what you can think about is that assuming Gaussian process, what that actually implies is that we can use the covariance matrix to borrow strengths. So you can design the covariance matrix in a way that using a function of the village location as a function of village location, and it will respect what I've discussed before, that is, you give higher weights to villages that are close together compared to villages that are far away from the other. And we parameterize that using the variance, variance and the scale parameter phi there. So if you look at model 2, it has two components, S0 and X1. You can think about S1 as because S0 is common to the three models. S0 is common to the three models. And that's a way of borrowing strength across correlation across all the outcomes. And then we have X1 and S2 in equation 2 and equation 3. You can think about that as just the extra variation that is not explained by S0 in the model. So we have a scaling parameter, which is just saying what's the proportion of this process that goes into S2 and S3, Y1 and Y2. So these are model structure. Let's look at the way we have estimated parameters of the model. It turns out that this model has probably about 10 parameters, no more than that. So we've got a lot of parameters here. What we've done is just to use multiple marginal likelihood based on importance sampling to estimate parameter. Of course, you can choose any inferential method that you know, or this is what we've chosen here. The likelihood for the likelihood function here, you can see that that's in blue, that's the integral, it's intractable. So definitely you have to find a way to do that. So we use, and it can be expressed as expectation. That's just importance sampling. And then the way we've done that is just that likelihood function, you can maximize the log likelihood, which is the last equation that I have there. I don't have the pointer, sorry. The last equation that I have there. The main ingredients in estimating this parameter is that we must be able to sample the conditional distribution of the random effect given the data, which I denoted as W given Y. That's the main ingredient we need to perform inference on this. And in doing that, we use Metropolis adjusted MCMC algorithm. That was what we used. And if you're familiar with this approach, with this method, it uses the derivative first and second derivative, so which is good at it's just S in convergence to the posterior mode, basically. So these are the parameter estimates. The betas there, they're just this is the first parameter that we all know, which is just the relationship between those predictors and the outcome, or the mean, or the posterior. Yeah. And then I think what is important here to explain is the so I wanted to put the phi naught. So phi naught is what I wanted to make blue there. Sorry. So that what is important there is the phi naught, which is 5.76 there. So that phi naught is the scale parameter for the S naught that I discussed, which is common to all the three outcomes that I showed before. So that phi naught basically is just telling us the scale of the correlation. And then there's something called practical range. Because I'm using an exponential correlation function here, you can just multiply that 5 by 3, which is about 16 thereabouts. So you can think about that, this 16 kilometers, that is the extent to which you can do reasonable prediction. That is, villages that are at least 16 kilometers within your sample villages, you can make reasonable prediction. But outside that, the prediction you'll be making will be so uncertain. Let me just put it that way. You'll be unsure of the predictions you'll be making there. So, and then we have 5, 1, 5, 2. Those are just scale of the formation of the parameter. So how do we make predictions? We've only estimated parameter, and then we use plug-in prediction. And what's plug-in prediction? Just the parameter you've estimated, you plug it into our predictive distribution as if you know the parameters, and then you draw your predictive targets. So as I've always quoted, the answer to any predictive problem is predictive distribution. So our predictive distribution is given in that equation there. And the way you sample that integral is just you sample W given Y first, then the condition on the W, and you sample W star. So that integral looks complicated, but it's very simple to sample, basically. And that's how we sample our. So think about W star for the created grid on the area that you're considering. So W star basically it's a random effect for each grid. You are sampling, so you can sample for each grid. That's the new star there. And then when you have the random effect, just add the fixed effects that you already know. You know the data, you know the covariate that you're using, add them together as you have the structure of the model, then you have the predictions. So the problem doesn't end in just predicting. What do we want to predict? Remember, what we want to predict is safety of a particular breed or particular area, if it's safe for NGO or not. That's what we're interested in. And the definition that was given to us by the expert in this field, they said, okay, an area could be considered safe if the probability that the proportion of individuals carrying at least 20,000 microfilarial density is at most 1%, greater than 0. That is, if you're very sure that the people in the area, they are less than 1%, less than 1% of the people there are carrying 20,000 microfilarial density, then you see that area is safe. That is what it told us. And then we formed that mathematically. So what we did there is that, you know, if I've created a grid of 2 kilometers by 2 kilometers, then I can compute essentially the probability that an individual is safe at a particular location, which is the question I have there. This is just, can you see there to compute this? So, I just put the question there, but computing this is very easy, basically. And then T there is just because we're not only interested in the grid 2 kilometers by 2 kilometers, we are interested in probably the pseudo-EU that I showed you before, or you can also think about the department or the district that I showed you before. So to calculate that, then we included M of X, and that's the population density. So we try to compute the weighted proportion of the population that are infected with at least 20,000 of microfilarial density. That's the T integral. Then you're just waiting what we got from each grid, and then you cannot compute define if an area is safe or not using if the probability. So you say if T is less than 0.1, it's greater than 0.95, and safe, and then you can also declare if it's unknown. So basically, I want us to pay attention to don't know area. So we have safe, unsafe, and then we also define don't know area. Don't know area, those are areas of high uncertainty. And I'm going to show us in a minute what we did with that. So we have a way of defining safe, unsafe, and don't know. Yeah. But so the way we approach the problem is that we can actually do better. You've asked us the question, but we can also at least perform some simulation, look at scenarios. So what happened is that we use both data sets, lower scope and antibody data sets. We could also use antibody data alone, because you have a model that will trust now. You can use antibody data alone. It's just that the predictive equation distribution that I showed you earlier will change a little bit. You can see this equation here have changed. But we can, even if you give us antibody data alone, we can actually use it. We assume that we know all the parameters we've estimated, then use this integral here to make prediction. And then we now propose a two-stage strategy. And what that is that we are trying to help them say, okay, what you will do is that you will give us a limited number of data sets. You go there, probably just collect few data sets, you send the data to us. We develop the model, we classify the area for you as safe, unsafe, and don't know area. So the don't know area, that's where you go to use the lower scope. which is limited because you don't have enough, a lot of lower scope instruments. So you can go to the don't know area to use lower scope. So instead of we're just trying to help them to reduce the number of lower scope that they could use to feed. That is, just collect limited number and send it to us. They don't know how you go back there with lower scope. So that's the two-stage strategy that we proposed in this method. So let me quickly show us some results. So the first map on the left there is just when you use the data of lower scope and antibody use post data. That's the prediction. This 2 km by 2 km grid using the data that I showed us in the map. So you're able to make this prediction. The middle one, if you use antibody data alone, and the last one, if you use the two-stage strategy, we pretended that using our data that we try to classify as the training and test and think about it that way. We pretended that we don't have all the data sets. So what is important here to notice is that the left panel and the right panel there, they look very similar, at least they need to be similar, but you can see a lot of uncertainty in the middle panel, which is the antibody one. So when you use antibody data alone, you have a lot of uncertainty areas where you don't know if you're safe or unsafe. So we perform the same experiments at the pseudo EU level. Now you know what the pseudo EU, they're just my partition that I did the other time. And then we also do it at department level, which is the districts as they have it there. So let's look at this map. Basically, you can see that we have the same classification for the left panel and the right panel. So if you have both data sets, if you have gone to the field, use those instruments to collect the data set, or if you have used our two-stage strategy, you have the same classification essentially. And this is actually showing the advantage of our two-stage strategy and the advantage of using statistical approach, which you can use limited data sets to make reasonable inference. So summary, and we perform simulation, but I'm not showing the results here because I know my time will not be enough, so I've removed that slide. The two-stage strategy reduces time and cost, definitely. And then when we did the simulation, we found out that we can actually reduce up to, we have a reduction of about 50% lower scope that is needed to carry out the same exercise, actually. And then in our simulation, we actually check, what do you need to do? Do you need to sample more people or sample more villages? So we found that sampling more villages, visiting more villages is more beneficial than sampling more people. And this work, there's plan to also extend it to other endemic countries. These are some references, and thank you very much.
Thank you. Do we have questions for Usutu? Maybe I'll start with one. Sorry if I missed this, but you mentioned places can be determined as safe or unsafe in mass drug administration. What actually would make it unsafe? Is there risks associated with the drugs that are provided for them?
Yeah, maybe I can say that in my introduction. So the informative tablets. So what happened was that I think when they were administering it in unsafe areas, and unsafe area means where you have high risk of lower. So on your safe areas, they found out that those people are developing some kind of severe adverse events. So that was what happened. That was why we actually said we should do this so that they could rule out some areas where they won't go with mass drug administration. And what they would do instead is that I think test and treat something. Is it test and not treat? Yeah, test and not treat approach. That was what they do. So they test before they treat you, basically. That was what they are doing in your safe areas.
Any questions in the room? Yes, let me bring the microphone up to you at the back. I'm here now.
Thanks. Thank you. It's really interesting. I love these types of studies. ENVI was environment.
Yeah,
So-
enhanced vegetation
index. So you're taking samples within villages and you're combining it with some of these environmental sort of things, altitude and what about scale? I mean, if you could have several villages within a district. So if you sample at a very local level, scale is different, but then use vegetation index and then you're aggregating things, right? Is the scale between the different model variables, are they different or are they sort of, is it accounting for things like that, differences?
So if you're asking the scale,
Yeah, and just in the also when people yeah, and the disease or like
Yes, yes, yes. Oh, thank you very much. I think that makes sense. Yeah. So basically, we have the village location. That means we know the coordinates of the village. So we can essentially just extract the predictor for that location, and that's what goes into the model. So the question will be the variation of those environmental covariates in space, basically. At what scale do they vary? And which is a good point anyway, yeah.
Okay, are there any final questions for Suji? Yes, I'll bring my phone on to you.
Thanks. Kind of a bit similar to the last question, but you were given a statistic of being 95% sure about something. And I was just wondering if that's sure enough, because obviously that means you're wrong 1 in 20 times. And if that is catastrophic for those people, do you need to be a bit surer than that? I don't know.
Thank you very much. That's a very good question. I think I've received that question somewhere else as well. I think, so the answer that I always give is that this definition of safety, I didn't come up with it. So the experts give us this definition, basically. So, and I completely agree that probably we can increase that to 0.99 or something, but yeah, but the definition was given to us after several discussions, several meetings. That was why they agreed this definition. Even, you still have people that didn't agree with the definition, but at least there's a consensus around this definition.
Okay, let's thank Olatunji once again. And now I'd like to introduce our final speaker for today, Dr. Claudio Frontere. You just pull up his slides. So Claudio is a lecturer in biostatistics at Lancaster University within CHICAS, the Center for Health Informatics, Computing and Statistics. He's a member of the Neglected Tropical Diseases Modeling Consortium, and his research covers spatial statistics for real-time health surveillance in low-resource settings. So I'm going to pass to you, Claudio.
Thank you very much. All these. So thank you for the introduction and the invite. It's a pleasure to be here. You're going to see some similar ideas to what you saw before in Olatunji's talk, but applied to another disease. And the main idea is again to combine data, prevalence data that's being collected using different approaches and different diagnostics, with a focus on another disease, in this case, lymphatic filariasis. I'll start with a brief introduction on the left, because I'm not sure if everyone in the room is aware of this disease. Is one of the many neglected tropical diseases that I work with. It's a vector-borne entity, and it has been targeted by the WHO for elimination as a public health problem by 2030. The WHO has these different types of elimination criteria. When they say elimination of public health problem, it means to lower the prevalence down to a certain level that doesn't cause a lot of morbidity. And the next goal would be, of course, elimination of transmission. That means to bring down the prevalence to an even lower level at which the vector cannot transmit the disease anymore. But the goal for 2030 at the moment is for most of the countries that are currently endemic the disease to achieve elimination as a public health problem. The disease is transmitted by 3 different species of mosquitoes, Aedes, Anopheles, or Culex, based on the specific area of the world that you live in. And what this disease causes is some disruption in the lymphatic vessel that, in the worst case scenario, can lead to very disturbing phenomena like adversarial lymphedema or elephantiasis. And on the left, this is a picture of the parasite that will navigate within our blood. On the right, this is an example of a person affected with elephantiasis. That is one of the most extreme conditions that LF can lead to. And a little bit about the life cycle and the transmission cycle for this disease, because we need some of this information for the modeling approach that we're going to use. So, of course, everything starts with an infected mosquito that will take up a blood meal from the human, that is the host, transmitting the larvae into the blood. then they navigate towards the lymphatic system. And there they start, of course, causing an infection and some of the diseases that I mentioned before. While in the lymphatic system, they start reproducing, and they become adult worms, then they start reproducing, and they produce some offsprings that are called microfilariae. And these circulate during night in the human blood. And those are the ones that then get picked again from another mosquitoes that then brings on the infection cycle. This map shows a little bit of the spatial distribution of the disease and the area of the world where you can find endemic countries with lymphatic filariasis. It's not a case that afflicts the tropical areas of the world, because those are the type of environmental condition that the vector needs to live, and also affects the most poor areas of the world as well. So a little bit about prevalence surveys and decision making. The WHO helps, the World Health Organization helps a lot of countries conducting monitoring and control programs to assess what is the current status for the diseases and how to treat the areas in the most efficient way. And the very first stage of these control programs is to do something that is called mapping surveys. So essentially, these are prevalence surveys where the minimum amount of data that collected is this triplet, where we have the location X, where the survey was conducted. This could be usually a community, a village, or a school. The number N of individuals tested, and then Y is the number of individual positives out of the one tested. And these surveys are conducted before doing any treatment at a sort of baseline status to assess what is the current burden of the disease, the current endemicity status. And based on that, then there are different prevalence thresholds that will provide some advice on what is the appropriate level of treatment. So should we treat that area 2 times per year, or should we increase or reduce the frequency, or is that a non-endemic area that we can leave untouched? And these also serve to kind of understand both the burden of LF, but also its spatial distribution, because remember that we usually work in very low resource settings, so we want to do spatially targeted treatment and use our resources in the most optimal ways. Now, when it comes to conducting these mapping surveys for LF, historically, 2 types of different diagnostics have been used. The first one was conducting parasitological surveys. So that essentially means that you need to take blood samples from individuals and you count microfilariae under the microscope. Now, if you remember, the MF circulates in the blood only during night. So these type of surveys are kind of very costly, time consuming. They have to be conducted during night, and then you need microscopes and expert people that look at the blood films. Around the year 2000, then they developed a rapid diagnostic test that is an antigen test, the ICT. So this is a serological approach, and this essentially detects the presence of adult worms circulating in the blood. So what happens was that until 2000, we had a lot of surveys that were parasitological surveys that measure MF prevalence. That is also the main indicator and what has been used to take decision on. And then they switch almost totally to serological surveys. So we have a situation where we have a very big amount of survey conducted with the rapid antigen test that measure antigenemia prevalence, and a smaller number of surveys that measure MF prevalence. That is what we're interested into. So our goal essentially is, are we going to use this abundance of ICT data, ICT prevalence data, and combine them with the less abundant MF data to come up with a model that is able to predict the distribution of MF prevalence? And to do that, we're going to exploit 2 things. So the first thing is that although this diagnostic measures slightly different things, those 2 types of prevalence, they are correlated. So we want to exploit that level of correlation. And the second thing is that these data usually have come with a certain level of spatial correlation. The reason for that is because the vector itself has a very peculiar spatial distribution that is determined by environmental condition. And those environmental factors like temperature, rain, humidity, and so on, they don't change abruptly, they change smoothly. So if you sample two villages that are very close to each other, it's very likely that they will have a very similar level of prevalence due to the similar vector distribution in those two places. So we can start by assuming that those data essentially come from the realization of a monomal distribution, okay? And we could start by using, for example, a logistic model, but then we need to come up with an approach to link those two prevalence, the ICT-MF prevalence, and to try to also factor in the fact that things are correlated in space. A little bit now about the modeling approach that we used. So if I define with W the number of adult worms in a sample individual, and then with M the number of offsprings that are produced by this adult, I will start with two very simple assumptions. So I will say that W is distributed as a Poisson random variable with a certain mean lambda, and lambda is the intensity of the worms in a certain individual. And then conditionally on the number of adult worms that are in your blood, and based on a certain reproduction rate that I'm calling alpha here, then the number of offsprings of children will, again, we can assume that that is a Poisson random variable with mean equal to alpha times W, that is the number of worms. Now, with these two assumptions, I can work out what is essentially the probability of testing positive based on which test I'm going to use. If I'm going to use the rapid antigen test, then that probability is simply the probability of having a positive number of adult worms in the blood, okay? And then we don't care too much about the math and how we arrived here, but it's simply a way of starting from those two distributions and make the right calculations. And I also factor in the fact that this rapid antigen test is not perfect. So it has a certain, the specificity is very high, but it has an imperfect sensitivity. So I also factor that into the probability of testing positive. And then instead, if you conduct a parasitological survey, so if you're counting offsprings MF in the blood under a microscope, then you will test positive if you have, of course, a positive number of those MF in the blood film. Now, these allow us to then put those two probabilities within our binomial model, okay, and exploit the fact that those two prevalence are correlated and are correlated through this parameter alpha and this parameter lambda. But there is still a limitation that we're making the assumption that the worm intensity, okay, is constant over space, is homogeneous over space. And we said that we want to exploit that these type of data are spatially correlated, and the worm intensity, how many worms you will find in a certain person, will depend on how much transmission there is, and that probably depends on environmental factors and other characteristics. So what we do is we extend the model by allowing that parameter now to be a function of space. Okay? And we do that on the log scale because that has to be a positive quantity. Modeling that on the log scales guarantee us that that quantity will stay positive. And the way we then factor and specify now this spatial intensity, spatial level of worms, is by using these three elements. So the first one is the spatial variation that can be explained by known factors, like alternative presentations, environmental factors like the vegetation indices, rainfall, temperature, humidities, and anything we know that is related with the distribution of the vector. A lot of time we're not either able to measure those factors, or we don't know what other factors are there that influence the distribution of the vector. So what we do to capture any residual spatial variation is then to add the set of random effects that are called S, that are just a spatial Gaussian process. So it's a set of spatially correlated random effects that will capture any residual spatial variation that cannot be explained by known environmental factors. And the last term, Z, is another set of random effects. This time they're independent, so they will capture any a residual variation that is unstructured, okay, that doesn't have a spatial structure. So this is our final version of the model. The other interesting thing is that by calculating these two prevalences and putting them together, we can actually work out what's the actual relationship between the ICT prevalence that we have on the x-axis and the MF prevalence on the y-axis. And that the particular shape of that relationship is going to be controlled by that reproduction rate that we're going to estimate from our data. The only thing that I'm going to keep fixed and known is the sensitivity, but all the other parameters in the model, so this reproductive rate alpha, the regression coefficient beta that tells me about the direction and the strength of the relationship between the environmental factors and the worm intensity, and then all the parameters that govern the random effects. These are all things that we're going to estimate from our data. The interesting thing is that by making sensible assumptions based on how the offsprings are generated, we come up with a type of relationship that is what is actually known in practice. So we know that ICT prevalence tends always to overestimate MF prevalence. That is what we would expect. And then we can use this to convert between the two prevalence. Now, I won't talk about too much about these. So essentially, when you use spatially correlated random effects, you need to make some assumption on what is the correlation function that you're going to use. And there is this wide class of correlation function called mater correlation function that are pretty flexible, where the mean idea is essentially, as Olatunji was saying before, as the distance increasing between any two survey location, then the correlation between them decreases. Okay? And the strength of that correlation is governed by this parameter phi that we estimate from our data. I will skip the details about the inference. Essentially, we use a likelihood approach, a frequentist approach to estimate these models. These models are quite complex, so the likelihood is not available in closed form. You have a multidimensional integral that you need to approximate somehow. The method that we use is Monte Carlo maximum likelihood. It's just a way for approximating that integral. And but you could replace that with any other inferential approach that you have tested and you know that is robust and you can rely into. If you want to know more detail about the inferential machine that we use, I'll be happy to talk about that later. But I want to move to an application. So here we use some data from West Africa. These are baseline surveys conducted between 1954 and 2012. So these are all data collected before any treatment was done. So they kind of represent a sort of endemicity equilibrium that was reached over a long time of period. And the green dots, they are the parasitological MF surveys. So you can see there are much less of them, around 300. And then we have much more abundant surveys that were conducted with the new serological approach. So antigen prevalence surveys, almost 1,000 surveys. And on the right, this shows a little bit that the spatial distribution of prevalence measured by the empirical prevalence measured in those surveys. So what is, in our case, our prediction target of interest? The nice thing about using these geostatistical models is what we get out of them is not just a point prediction, okay, just a single value. What we get from them is the full predictive distribution of, in our case, what is of interest is the prevalence of microfilariae. Okay? And then we can use that joint probability distribution to calculate any summary statistics that is of interest. That could be the mean, could be the quantile, could be the probability of exceeding a certain prevalence threshold. More importantly, since we can calculate that predictive distribution at any location, so any unsampled location in space, but treatment decisions are not taken at a point or at a pixel level. They're usually taken at some administrative unit level that for lymphatic filariasis is called an implementation unit. So the real target of interest here is the population weighted prevalence of MF at implementation unit level. And we weight by the population because you can imagine having this model that also exploits environmental factor. You might predict a very high risk for LF in an area that is highly suitable for the vector, but no one lives there. Okay, so you want to weight your predictions when you aggregate them at implementation level, considering where people actually live. And since the interest is in eliminating diseases as a public health problem, the threshold that the WHO uses is 1%. So if prevalence goes below 1%, then we will say that we can eliminate LF as a public health problem. Then this threshold might change slightly based on the type of vector that is transmitting the disease, but these are all inputs that come usually from WHO guidelines. And we are not going just to look at if it is above or below. We won't, since these models are not perfect, of course, and we need to consider that we work with a small sample size and we don't have a census of data. Then we also need to factor in the uncertainty from this model. And in our opinion, good ways to provide what is the probability that your target prevalence is going to be below your threshold of interest, okay? And then this level Q that in Olatunji's presentation was 6 to 95% is again a decision that the policymaker has to make, okay, based on how much they are risk adverse and how much are they worried about committing errors in one direction or another. So if they're worried more about over-treating or under-treating certain areas. So this is usually not a statistical decision, but it is a policymaker decision. I'm going now to show some maps from fitting the model that I introduced to those data. On the left, you can see pixel level predictions. Those are 1 kilometer level prediction that are showing the predicted average, the predicted mean prevalence of MF. So by exploiting both ICT and MF data, then we can predict what the prevalence of microfilariae is at any desired location. Of course, then we can aggregate on the right at implementation unit level, and these maps could be used already to make decisions, but what we suggest to do instead is to look at the probability of exceeding that threshold, okay? So you will, in this area that are red, where that probability is very close to 1, then it means that you're pretty sure that you're above that threshold, okay? So you need to keep treating. While you have other areas here in blue, where you're very sure that it's probably a non-endemic area and you can stop treatment or you can start surveillance post-elimination. And the more you get close instead to 0.5, that is the highest level of uncertainty. So those are probably areas where you want to go back in and conduct more surveys to understand what's going on. So let me draw some conclusions. So I think it's important when we have information about the underlying nature of the process and the biology and the transmission process to try to include them in our model. Okay, that's why I call my model semi-mechanistic, because we use some understanding about how the adult worms reproduce within the body to build those models and those probabilities and link them together. Of course, this is not a free lunch, and I really like this citation from Combs that say that we buy information with assumptions. The consequence of that is that if our assumptions are wrong, then what we're buying is bad information. And so we also need to be careful where we include these assumptions that come from scientific knowledge into our model, how much we're confident about those assumptions. Because if they're wrong, then we'll do more harm than simply using more empirical approaches. The results from these models, of course, can be used to assess which areas need treatments and to come up with more spatially targeted intervention. And another use of this type of output that we're actually done already is to combine these sort of pre-intervention results and predictive maps, feed them to mechanistic models of transmission for lymphatic filariasis, and then from them, we can generate scenarios where we understand, okay, where we're going to be in 5 or 10 years based on different treatment scenarios and frequency of treatment. So this is another useful use that can be done with the output from this type of models. That's all. I'm very happy to take any questions. Thank you very much.
So I'll ask, do we have any questions for Claudio in the room? Yeah, I'll come over with the microphone.
Thank you for a great talk. I was wondering, because you're using surveys spanning from the 1950s to more recent periods, whether you try to, could some of the way data was collected change, or because you're assuming, for example, the sensitivity is the same for all the source of information. So have you considered that, or maybe have you tried different time periods? And then I have another question.
Yeah, so we did two things there. So we tried to look first if there was any temporal variation as well. And yeah, thank you. So we tried to look if there was any temporal variation in this data. We didn't find any temporal variation. That kind of reflected the fact that they were representing a sort of endemicity status before treatment. And we have partial information how the data were collected. We're not too much worried about the ICT data because the type of diagnostic tool that was used is essentially the same from the same producer. There might be maybe some slight difference in the data that were collected through a parasitological approach, but that is a type of variation that you can always have, because if you probably ask different individuals to look under the microscope based on their skills and everything, you can have slightly different results, and the count of MF might vary. But yes, those are not things that we know and that we can include in the model, unfortunately.
Did you say there was another follow-up question?
Yeah.
I'll bring it over.
My second question is, in all the maps, the results you showed, there seemed to be a clear divide between the north and the south. And I was wondering whether there was a reason, a natural, maybe a river or a natural reason why there was such a difference between the north and the southeast.
Yeah. The honest answer is I don't know. I don't know why there is this neat difference, but I mean, it's essentially coming from the surveys, and for historical reason or another things that might be going on a lot of time is that although these are survey conducted before any treatment for LF was done, it might be that some areas, I mean, two things could happen. Either there are different environmental conditions that might explain those big difference. But if you notice, they seems like almost at country level, those differences. So another explanation might also be that while they were not doing any MDA treatment for LF, there might have been other entity programs going on doing treatment, like for onchocerciasis. Yes, I'm trying to thinking about other diseases where you have where you provide drugs that might partially affect also the distribution of LF, but that might be an explanation, to be honest. Yeah.
And do we have any other questions for Claudio? No, no questions online? No. Okay. Well, in that case, let's thank Claudio and all our speakers once again. And I'd like to say, yeah, thank you very much for joining us for these really interesting talks this afternoon, whether it's been here in person or online. In person, we do also have like plenty of tea and coffee over there and biscuits as well. So please do help yourself. Just make sure you leave the cups behind. Thank you.